I don't think Loic needs much of an introduction. Like I said, a lot of us at Janelli already know him. He's right now a group leader at the Quantitative Cell Science at the Chan Zuckerberg BioHub. And I'll turn it over to you.
Thanks a lot, Bill. Thanks for the organizers to invite me to give this talk on scientific visualization. Let me share my screen now. So today, in the context, so visualization is something I've always been quite passionate about. I must say, ever since I started my PhD, I was always interested in images, in ways to communicate visually, and I think that's probably one reason why actually I transitioned from pure bioinformatics during my PhD to microscopy. Which was kind of radical transformation, but a very enriching one. And I was super lucky to actually, join Jean's lab at Janelia and get exposed to fantastic research there, fantastic microscopy, and I learned everything of the microscopy there with people like Philip Keller and others. And so very exciting to give this talk here to you guys. And so. Really, what I want to talk to you about today is Napari, which is really started as a dream, as I will explain, and really became something that has touched many people and is being used by many, and it's actually pretty amazing to see how a small community grew very fast and made possible all the things that now are part of Napari and all the things that have been enabled by Napari. So. Let's get started. So first, as a little bit of introduction for myself and my team. So I work at, as Bill explained, I work at the Chan Zuckerberg BioHub. It's a non-profit research institute here in San Francisco. It brings together UC Berkeley, UCSF, and Stanford University. But we also have intramural research program of which my team is part of. And currently, I'm actually hiring an optical engineer if someone is interested. Actually, two of them. And so my team is really trying to do something special, which is trying to combine both the design of state-of-the-art algorithms for image processing and image analysis, but we also design and build logic microscopes, following the tradition of all the things I've learned back in Janelia. And we try to combine all this technology to basically try to answer some biological questions. So my goal for my PhD, if you want, here at the BioHub is to turn into a biologist slowly but surely. And so using all this technology that I've learned in the past years, and I've been developing and pushing it further, and then really trying to answer questions with that. So that's really what my team is about, just to give you a bit of context there. But of course, visualization has always been an integral part of that ever since I started playing with microscopes. So we do actually build these machines, right? And these machines produce a lot of image data. I'm not gonna explain that to you guys. And this data are images. And so once you finish acquiring these images, you need to visualize them, right? And so we build, kind of a sim view that is kind of the following the Keller lab design that I also participated in by writing this autopilot system that you might know about. But here we also have our own kind of California version of SimView, which my team has been building and using for doing some interesting biology. We also have built some new kinds of logic microscopes based on the single objective design, and we've been pushing resolution, field of view, and speed there. But as I said, so once you have built these machines that can produce gigantic amounts of data, tens of terabytes per day easily, and if you have multiple machines, then it starts to go to be huge. You need to also be able to look at this image data, visualize it, and even if you don't go into the large scale data sets, even if these are small data sets, you still need to be able to conveniently look at it. And so in the past, I've worked on things like, for example, Clear Volume. That was a kind of Fiji plugin to visualize data in 3D using fast GPU-based, volume rendering. Started also as a weekend project, similarly to Napari, and it got people excited. We brought the team together and we pushed that, and we could at the time do things that were quite fun, like for example, do live visualization of light sheet data in the cases of kind of fast acquisitions. And so we could here, for example, image, developing cell against embryo at 6 volumes per second and visualize in real time, how the cells are dividing and kind of how the embryo is forming. So already then, back in 2014, 15, I was always thinking about how to look at this data in novel ways. And so with Philip, we were, of course, work on this autopilot project. We also had to visualize things. So here, for example, some stacks that are rendered with clear volume at the time. And so really, another thing that kind of exposure to light sheet and kind of advanced bioimaging was like, you really need to look at the data in 3D. And so that also informed a lot of decisions for Napari going forward. And you'll see that in a second. So here at the BioHub, we also acquire images on our California SimView, and this is an example of a 2-color recording. This is also rendered with clear volume in this case. This is kind of already an old video from us. And this is work from Merlin Langer and Giorgio Bonagantini in our team and others. But I mean, what happened when I started my lab in 2017 is, in San Francisco is that there was a dramatic shift in the way, in terms of the languages, the programming languages that were used for scientific computing. And there was really a kind of, I really think 2017 was kind of, for me and perhaps for others, the watershed moment where really I saw that there was really a big wave of high quality, highly performant and impactful scientific libraries in Python. And everything I had done before was in Java. And I suddenly felt that there was a bit of disconnect between where I was with the tools I was using and where the field was moving. And not just bioimaging, but I really, because of my exposure to the Bay Area and a lot of people here that were actually working with Python and developed some of these libraries, I really realized that the future of scientific computing beyond just bioimaging was definitely moving towards Python. And so I thought that maybe we needed to help maybe this transition. And so, of course, in Java, on the basis of the Java programming language, there was already a fantastic and really feature-rich image viewers such as ImageJ and Fiji and also IC and extremely successful projects, extremely popular with lots of plugins and lots of applications built within and from the outside out. So really fantastic work there. But as I was transitioning to Python, and really as I decided, okay, now I'm going to learn Python, right? Because this is, I already had programmed things in Python. I was already doing some visualizations, but I had not really made the fundamental step of saying, okay, Python is going to be my primary language, and now I'm going to do all my scientific computing on the basis of Python and develop all the tools on the basis of that. So, as I made that choice, I realized there was no good image viewer in Python. There was nothing that was really good, and so. At the time, and it was really a kind of an interesting combination, very fruitful accident. At the very same time, I was thinking about this. Juan Núñez Iglesias, my good friend from Janilia, was actually visiting me. He was staying at my place, and I actually was at a hackathon for Scikit imaging in Berkeley. And then we realized that we had the same opinion that basically there was some big gap within the Python ecosystem. There was no really high performance image viewer that was really built for Python and that was really offered a set of features such as n-dimensional capability built in not a web browser app, because we really felt that we wanted something that didn't have the indirection and kind of the layering of a browser. We wanted to have a first-class application, or at least something that felt immediately accessible. We wanted something really fast, so GPU acceleration was definitely a must for us, and especially for me, because I had worked with clear volume and I knew that you could get a lot of performance there. And we had some ideas along seamless integration of 2D and 3D viewing, and we also wanted to go away from this notion of having lots of windows popping up on your screen with lots of images, and we wanted something a little bit more integrated. And so we took inspiration from other tools that had been developed for almost decades, such as Photoshop and Illustrator, and we're thinking along these lines. And this was really a dream at that point, right? So this was a weekend he was spending at my place. We were dreaming about all this, and we actually realized also quickly after that there were other people in our sphere there here in San Francisco, such as Nick, but also people like Jeremy Freeman. There was also an Janilian. They were also interested in these things, and so we started this project, Napari. And the reason we called it Napari is because Napari is an island, or technically it's a city in an island, that is between San Francisco and Melbourne. Melbourne is where one lives, and San Francisco is where I live. And so we decided, it was really literally, we created the repo on the train going to the Computer Science Museum down south. And then, while creating the repository, I was like, I had my laptop on the train. I was like, okay, we need a name for that thing. And in 10 minutes we had a name, and that's it. And so we started writing code in the weekend. There was actually at that hackathon, there was Kira Evans that one had noticed, who was extremely a talented programmer, contributor to Secret Image. And so we basically, so I decided to hire Kira and for an internship. And this is how things started. I had Ahmed Shani in my team, who was actually a software engineer in my team, who also started coding on Napari. And then a bunch of people at the BioHub started to join, such as Kevin Yamushi, who's now in Europe, Bryant, who's moved on to another company here in the Bay Area. And just step by step like this, the team grew up. And then, Nick started to also put a lot of work and start to organize the community. He really took on the project as kind of to manage it on a daily basis, and has done an amazing work pushing this project forward. And I think we really needed a full-time person, someone who would really take the project in his hands and push it forward. And so this was the start. But then, now the current situation is we have people like Tali Lambert, who in the past year and a half has put has contributed amazingly, and has put a lot of work in this project. And we have many contributors, and it's impossible to mention them all by name, but here's the list. And we have a lot of kind of core developers now, more than 10, I think. And it's just increasing regularly, and we have more than 100 contributors all over the world. We have three meetings in 3 different time zones for developers, and for people to kind of meet and contribute and kind of exchange ideas. We have different working groups on different aspects of Napari. So the project is grown much further than I could possibly imagine with one, and this is an amazing. I just want to thank the community for making this dream really true, and it's just an amazing outcome. So what is Napari? Let's look in a little bit more in detail. So as I said, it's a Python library that you also can think of as an app. Here, we have a standalone app, but primarily it's a Python library for visualization of n-dimensional images. It's GPU accelerated, so it's very fast. It integrates very well with the whole Python ecosystem, so with NumPy, with Dask, with ZAR, with Qt. So you can do a lot of things. You can interface with a lot of existing libraries, and in a very natural and Pythonic way. It supports arbitrarily large data. There's some caveats in terms of dimensionality and so on. we can, in my team, we regularly browse terabyte scale data sets with Napari with a combination of ZAR and DASK. So all our data is stored with ZAR, and we actually load that through DASK, and we can basically browse the data set very conveniently. Of course, the data sets are oftentimes so huge that the limits are then the network, the speed of the computer, the size of the GPU memory on the GPU card, and other aspects. But if you have very powerful machines, you can actually do a lot of really good work with Napari, and it's extensible and customizable in many ways, and it's inclusive and community driven. The community aspect of it is very powerful because it means that the project is really growing at a rapid pace, and new features are added in a very organic but also very organized way. I mean, it is a challenge, and it's quite an amazing work that Juan and Nick and Tali have done in terms of like keeping the community organized. We have a whole a governance model for that. And as I said already, that we really rely on an incredible set of libraries that have been developed previously. And like early on when we were in this train, with Juan, opening up that repo, the first thing we looked at is what's the library we're going to use to build Napari upon, like what's the graphics library we're going to use. And we were thinking about OpenGL, but then we saw that there was this vSpy library that was incredibly powerful and incredibly well designed. And we decided, okay, vSpy is going to be the basis for Napari. And this was probably the best decision we could make, and has enabled a lot of features that Napari has not today. And I will go through some of these in the next slide. So the anatomy of the Napari viewer goes as follows. So we first have the canvas, which is a place where you actually look at the images, and you have a layer list, which means you can actually overlay different information in the canvas, be it 2D or 3D or ND. You can basically overlay all this information in the canvas, but the layer list controls the images, but also the other kinds of data that you can layer on top of images. And so this was a big feature for us, because as I said, we wanted to go away from this window explosion that I had seen many times on people's screens, where lots of windows open with different images, and so we wanted to avoid that, having a much more ergonomic approach to it. And so then per layer, we have layer controls that lets you control things like the opacity. In the case of images, you can control the min and max intensity, pixel intensity that is being displayed, and then everything else is clipped. We can control the gamma, which is very important because most screens have a random gamma that you might not really control, and so it's very important for I mean, something I was really pushing always for is to be able to control gamma, because that's oftentimes the best way to adjust the brightnesses to make them more accessible to our perception. Color maps, of course, blending, interpolation, all sorts of features that were really part of vSpy we could leverage. And of course, as in many other such tools such as Fiji, ImageJ, and IC, we needed these sliders, right, to be able to not just display 2 or 3 dimensions, but potentially more of them by actually slicing through the data using the sliders. And so some of the first prototypes I was playing with, like, okay, let's create a 10-dimensional NumPy array. Can I display that? Do I have all the sliders popping up? And so that's what this slider does, right? It basically lets you slice through the dimensions. We had some ideas of being able to do max projections through a double slider, and some crazy ideas, but there's some performance limits there. So we wanted to be even more innovative there, but there's been some limits, and we're I guess at some level still thinking about how to push the limits of all that. And of course, you want to be able to add and remove these layers, and so I will go into detail next about the different layers that are part of this. Finally, the different you can control the canvas in terms of switching between 2D and 3D, rotating the dimensions, flipping the dimensions, going from a mode where you overlay everything on top, all the layers on top of each other, or arrange the layers into a grid. And you can also go back home to reset the view of Napari. There's also, we actually have a Ipython terminal built into Napari, so you can actually write code within Python and interact with the layers. So a lot of cool features. What people do a lot also is actually open Napari from a Jupyter notebook, which is possible. And finally, the status bar, which is really where, as in many other tools, where you can see which layer you're looking at, the coordinates of the pointer, and what's the value of the pixel that you're looking at in the case of pixels. So as I said, layers are a very important concept in Napari, and we have lots of different types of layers, such as labels. We have point layers, we have geometry layers, and I will go into a little bit more details. For example, these are points. You can put points onto your canvas, and basically, this corresponds to literally a list of coordinates. And so here is an example. I don't know if okay, the video is playing. So here are kind of a showcase of all the kind of layers that you can have in Napari. So here I would like to say also, like, most of these slides I'm showing right now are made by Tali Lambert, who has shared these slides to the Napari community, and I cannot make them better. So I'm literally using them as he has designed them, because they're just amazing. So thanks Tali for sharing these slides with the community. So here you can see different kinds of layers showcased. And so we have image layers, we have labels, we have points, and because it's all kind of down below that is all OpenGL running and GPU accelerated, you can push a lot of geometry into Napari, so that you can have millions of points displayed. You can also have vectors, like what you see here on the bottom left. We also have a track layer, which lets you basically show the trajectories of objects in 3D and in fact in ND. We also have a surface layer that is also very exciting potentially, for people that do kind of functional imaging of different kinds in this case. So let's have a little bit of a more close look about the image layer here. So with the image layer, you can control different things. You can switch seamlessly between 2D and 3D viewing. You can control the field of view, so you can change the type of projection. You can slice through data when you're in 2D mode, but you also can slice through data when you're in 3D mode. Or if your data is 10-dimensional, you can display a 3D slice and then slice through it with all the slices you could possibly have. And because we have these different layers, you can actually do blending and actually do multicolor viewing of your data sets. We have, I think recently, we have new features that let you combine and split some layers on the basis of color. So in fact, there's so many new features at every release that I don't know them all right now. So that tells you something about how fast the project is going. And then another example is that shows kind of the interfacing of Napari with the Python scientific infrastructure. Here, by leveraging ZAR and Dask, as I've mentioned, we can actually access very large images. And in case of 2D, we really fully support image pyramids. So we can, you can here, for example, browse very large arrays and very large images. And basically, you have an image pyramid system that will basically make sure that you can access the data at all scales. Here's a very nice example of lattice light sheet viewing and also live computing with Napari with the help of Dask, which lets you build these compute graphs that can be run while you image. So basically, as you image, there's lazy loading and lazy computation of things like, for example, here, deconvolution. So this is actually not just deconvolution, the skewing, loading, the skewing, and deconvolution is happening live as you slide your slider here. So it's a very nice application from Tally Lambert on his lattice data. So in case of lattice data, it's the stacks are really rather small compared to some of the stacks that we actually work with. So this sort of thing doesn't really work for much bigger data sets, not because Napari dashboards have a problem, it's just because it's just that we don't have yet computers that are fast enough to do real time to do this kind of processing real time on images that might be 6,000 by 2,000 by 800 voxels or something, which is what we typically do these days. So definitely very cool capabilities, but we're almost at some point reaching the limits of what the computers can do if we want to use the full capabilities of these libraries in combination with Napari. Next, we can have a look at another kind of layer, which is the segmentation, the label layer, which is you can load your images and then you can say, well, actually, I want to annotate them. And that's one of the big things about Napari, and this was really pushed by Nick Soferniev at the beginning when he joined us. And one of the things he really implemented early on is with others is that really adding this interactivity to Napari. It's not just a viewer. You can actually use Napari to actually annotate datasets and add information coming from the user in an interactive way. So here labels are perfect for that purpose. Like if you need to label a dataset, for example, training some segmentation algorithm, be deep learning based or other, you need to do some manual annotation, some manual work labeling objects in their images. And so Napari is really good at this. And so here you see an example of two layers, an image layer and segmentation layer, and how the user can actually label the different objects in the image here. That also, one of the key things with Napari is that we really pushed for everything to be ND from the get-go. So, and this is something that Juan Nunes Iglesias really was pushing always, and this came also from his efforts to do the same thing in Scikit image. And so we really trying every step of the way, we're really trying to make sure that everything works as n dimensions. And for sure, 3D is the first thing that needs to work after 2D, and ideally it works for ND anyway. But here are examples of how you can do labeling in 3 dimensions on some brain data from the Allen Institute. So here's another layer, the point layer, and that can be useful for many purposes, of course. One possible application would be for a single molecule detection, super resolution based on single molecule detection, and so yeah, I mean, so many possible applications of this, and especially when you can overlay that on top of images, but possibly on top of other kinds of layers. And so you can see that there's all sorts of possible interactions that and visualization opportunities when you combine these layers together, or you can use them individually. So anything is really possible here. Another example is here to combine a 3D image, which is in this case EM data, some points in the points layer, and vectors. And here this is an application for 3D electron cryotomograms of HIV from and this is really some work from Alistair Burt at the EPS Grenoble. It's fantastic, he posted that on Twitter, and we were all very excited because it's a beautiful application of how you can combine 3 different kinds of layers to basically not only showcase the data itself, but also the result of multiple steps of analysis potentially, right? And then show that all together and in a very, very easy way. I mean, when you look at the code in the end, it's very little code to actually achieve that. And all these features kind of, cross-fertilize across the, like you can potentially imagine doing that also at large scale with very, very large images, and so on and so forth. Of course, there's a lot of work that we need to do to be able to be fully capable of rendering data at very, very, very large scales in a lazy way. There's a lot of complex programming and complex work there, but we're moving towards that slowly but surely. Another very important layer type, especially for our team who's really interested in tracking cells in developing embryos, is the track layer, which was developed by Alan Lowe. And this is initially developed by Alan Lowe. I mean, quite a lot of people now contributed to it, and also someone in my team, Joe Brigantini, has been recently making some improvements. So you can basically show these tracks and control the different aspects of viewing the tail of the track, but also the colors and different aspects of how the data is rendered. It's a beautiful way to render not just, for example, legit data, but also the tracks showing where the cells are going. And I will show you some data for this later. And in this context, Alan Lowe from UCL has made some very nice plugins from Napari where you can show the tracks directly in the canvas, but also he has added tools for basically showing the actual lineage tree of these tracks. And so that's really cool. One of the first cool applications of Napari, showing how you can add some additional panels to the Napari interface to show other aspects of the data that might arise from analysis. Finally, the last layer type is surfaces, which basically literally just triangle geometry. And you can basically, if you have a data set that is in the form of a mesh, so a set of vertices and edges and essentially triangles connecting all of that into a surface, you can actually render that in Napari, overlay additional information per node, and have automatically things interpolated. And then with that, you can actually do render that in 3D and also play that in time if you have time series, as you can see here. So really fantastic, fantastic capabilities. And all of these layers, they can all be combined together. So you can basically render meshes on top of images and have points and vectors and tracks, everything is possible. So it's just the only limit is your imagination, how you want to showcase the raw data, different steps of analysis, all the way to a final analysis and result. So this was a kind of an overview of all the layers. Oh, I forgot one, the shapes layer. There's another one, nice one. It's mostly 2D shapes, and you can basically, do more detailed annotations or drawing on top of 2D images, and that's quite useful for many applications that require very, very, very precise annotations. Yeah. So as I mentioned before, one of the key ideas of Napari is that you can also, so you can view the data, you can view the raw data, you can view these layers that kind of may be the result of an analysis. But you can also, because we have this Ipython terminal within Napari, you can actually run some of the code that will take the raw data and push it through different steps of analysis using libraries that you might be comfortable with already, such as scikit-image, scikit-learn, of course NumPy, and if you want to do deep learning, you can also use TensorFlow and PyTorch, and so you can take your raw data, push it through some analysis, and then display all the results of this analysis as new layers of different kinds. They could be images, but they could also be points or so on. But you can also, in addition to doing all of that, you can interactively add information in the canvas in the form of here, for example, adding points to mark certain objects, and then use the data that is that underlies the result, that annotation. Use that as part of your own algorithms, and as part of your own workflows, to be able to combine all of that, the raw data, and you kind of automated processing input coming from your own annotations, and run all this to get some new results. All right, so really powerful viewer, a lot of interactivity, and you can run your Python code from within the tool, or from within Napari, or from a Jupyter notebook, and basically get your whole pipeline really well integrated with a very powerful visualization and interaction to it. So this is really, I think, the value proposition, the main value proposition of Napari for people that do bioimage analysis in Python. Here's a nice example that one the nice way of kind of integrating some existing code or libraries into Napari. He basically wrote a kind of mini TensorFlow within Napari to show actually the performance of training a neural network. And the neural network in this case was some work we did in my team on denoising images using self-supervised learning. And so he can basically here visualize the training, the loss curve, but also the resulting images where you see the result of denoising these images. So very nice kind of little toy application that combines scikit-image, scikit-learn, PyTorch, Matplotlib. So really beautiful little example of that. But so at this point you might be asking, okay, so I want to try this, right, if you haven't done it yet. It's all Python, right? So you can install it with pip install Napari all. I really, recommend you check the website. There's a lot of documentation. There's been amazing work done by the team to populate, the documentation to make fantastic, fantastic work there in terms of examples and so on and so forth. I must say at this point, CZI has done an amazing work supporting the project through Nick Sofronev and through many other people that are working within CZI to support and to accelerate the development of Napari. This has been a great collaboration there and a fantastic outcome. And so there's not only Napari, the main website of Napari, but there's also the Napari Hub that you can find through the Napari website, which will list also a lot of other things, which are, for example, the plugins. We also have a bundle app, which you can try to be able to kind of run Napari as a standalone app. Although I think some of the thing right now is that maybe the primary way to use it in Napari, and maybe the best one is probably right now to actually use the pip installable in Napari. And it's maybe easier for kind of Python people that are familiar with Python to use Napari, but we really want to push forward eventually the use of an app. But that's going to take a little bit more time to get these things ironed out to the level where we can be confident that biologists, for example, or other scientists can really download Napari and have the full experience without any hiccups. So I was already hinting at the plugins, because this is the one thing that CZI has been really helping a lot with their Napari Hub website and other aspects. So part of the idea of Napari was not just to be a viewer, but we also decided we wanted to have the possibility to have a plugin, because this was something that had been successful in other platforms. And so we thought, let's try that. Let's see if we can actually help people ship their code to the users through Napari, using Napari as a platform, and not just a viewer, and not just an interactive tool for annotation and all the other things that can do. But and so Tali Lambert and others have really put a lot of work to develop the Napari infrastructure for plugins. So here are a few examples of plugins that have been developed. Plugins we have plugins for reading data from disk. For example, Napari ND-TIFFs, it lets you load different kinds of formats. And so here's an example of that. And of course, when we can use lazy loading, we do, and that basically also helps, the interactivity and the low latency when accessing, large data sets. Lots of cool plugins. The Omero team has also, developed a plugin with Tali. There's plugins to control microscopes already, such as this Napari Micromanager plugin. Robert Hazer, who's very active on Twitter, many of you might know, has developed a lot of cool plugins. That's one of them for his OpenCell-based image processing tool. Backlist, I won't try to pronounce it. So yeah, really cool, features are being added through these plugins, and very complex actually things being done here. You can see this very, very, very cool tool where you can actually build a pipeline for image processing, and then have it actually be exported to scripts that work also in other platforms such as Fiji or ImageJ, and even as Python scripts. So lots of very complex things are being developed on the basis of Napari and with the help of Napari that also interface and play nicely with other platforms. Salpos, I won't introduce it further. It's well known, and it has now for quite some time a Napari plugin. Stardist, of course, another very popular tool for cell segmentation, has also a Napari plugin, and works really well. I've actually tried it myself on some of our data. So other tools such as other works such as SelfFinder from Adam Tyson at UCL, and one really cool thing that Tali did was to actually add, to design this library, Magic GUI, that actually makes it possible for people that are not Qt experts. So Qt is a library for building these interfaces, these user interfaces. Tali designed this very cool thing, Magic GUI, that makes it easy for people to just write a little bit of code that explains how to connect certain parameters or functions to actually UI elements. And so basically, almost by magic, these very complex UI elements with lots of parameters get generated automatically for you, and so you don't even have to know how to do UI programming. So really fantastic, feature, because of course we want to facilitate the work of people to port some of their Python code and algorithms and workflows into Napari plugins, and so that was really a very great move by Tali to make this possible. Yeah, so here you can you can really see the interplay between the raw data, different layers, and the interface on the side, and how you can make all these things work together to create very interesting workflows and applications. One of our favorite plugins in my team is Napari Animation, which was started by Guillaume Witz at the University of Bern. Alistair Bird helped a lot there too, and Nick also put a lot of work on this plugin. And it's a plugin that lets you actually build animations from all the layers that you have in your canvas. And so you basically kind of define keyframes. You can turn on and off certain layers, change opacities, move the camera, change the point of view, and so by doing all of that, through the kind of animation wizard that you see on the side here, you can actually create pretty complex animations that benefit from all the features of Napari. And so this is, as I said before, like Alistair posted that on Twitter, and we were all super excited because suddenly all the things we could see now, we could actually not just see them ourselves on our machines, but we could then export them as movies and use them for our papers and share it with other people. So very powerful. So now actually we do all our movies for our papers using Napari animation. And in the case of very large light sheet data, we actually also can do that, right? So that has been, uniquely enabling, because the ability of Napari to interface with libraries like ZAR and Dask on one end and Napari animation all combined together means that now we can actually generate movies for terabyte scale data sets very easily. So easily that actually biologists in the team that are not programmers by training are actually able to do these things by themselves. And that's the place where I get really, really happy, is when non-experts start to use Napari effectively. And of course, they need a little bit of training. They need to be exposed to, it's still Python-based. There's still a few things that are still a little bit hard for non-experts. But I think, the path forward is really to iron out all of these things and make it every step of the way easier and easier to use. And I think we are doing great work towards that goal. So it's very exciting. In my team, we have also been using Napari as a library that you can use to embed a viewer within your own applications. So Anicha and my team has developed this tool island for self-supervised auto-tune and unsupervised image denoising. And this is really a standalone app that you can download, and it just works out of the box on all the operating systems. They really, there's almost, if you don't know it's Python behind you, you won't know, and there won't be any kind of Python problems that will cause you to realize there's some that is not that is Python behind that. So there's no updating, there's no environment, you just have a standalone app. And so if you want to do that, if you want to really ship something to users and make sure that it really works for them, that poses a lot of issues in terms of packaging and so on. But you can actually embed Napari within such a complex application that has a lot of constraint in terms of packaging and distribution and so on. You can still do that, and Napari works great. And it just offers a fantastic viewer within your application that you can use to view in the data, and you have all the benefits of Napari within that. Of course, I think our preferred approach is, of course, to have people ship their code through Napari plugins. That's the easiest thing to do. But in cases, there are some special cases where you really want to use Napari as a library within your own application, be it a very, very complex software for controlling macroscopes, be it some tool that you want to ship to users that know absolutely nothing about Python. So there are cases, but yeah, in general, I think our preference is for people to write plugins. But I just want to show that it's actually possible to use Napari as a library that will help build other things from it. And that's the power of open source and its modular design principles that are behind everything we do. Yeah, so you can do things like, for example, animations where you have two layers. One layer is the noisy image, and another layer is the denoised image, and you can just generate these beautiful animations by changing the opacity. And so everything we do now in terms of visualization, in terms of showing the results of our work, everything we do is based on Napari now. And so there are definitely applications in light-sheet microscopy, and the ability to visualize very large data sets and access them through ZAR and Dask is key. And in a recent paper, we've been using that extensively for all the videos in the paper. And this is our work on single objective light-sheet microscopy. So what we've done is, first, we use, of course, we have our own code for image processing and of light-sheet data that encompasses everything from registration, motion stabilization, registration, fusion, deconvolution, dehazing, intensity equalization, and more things. Diffeomorphic registration in cases where the things tend to wiggle too much. So we have all that, but of course, like you do all of that image processing at scale, we use Dask again, so we can run these things on clusters and process very large data sets. But we also need to look at it, right? And so our library DEX that we designed for all of that, which is open source and fully available for everybody to use, definitely uses every step of the way there's Napari to be able to look at this data. And it's also, we have a Napari plugin for it. But another fantastic thing is at the end of the processing, once you've done all that processing, once you have this ZAR file that is fully processed, then you can really visualize it with Napari. And again, with Napari animation, you can just render it on the screen interactively, or you can generate your MP4 for your publication. So these are actually videos that we generated directly from that. So you don't need necessarily some of these very expensive commercial softwares to make very complex animations. You can actually use Napari very successfully for all that. And here is kind of a little like I believe that's my last slide. Oh, it's not. This is kind of an example from that paper of how we can, overlay a lot of information with Napari and make really beautiful animations that showcase the work. And I think this is kind of a good example of layered visualization and processing and analysis that you can do with Napari and the tools that are developed in the Python ecosystem. So here you see a segmentation of a zebrafish embryo, and then interesting. The videos, for some reason, actually, let me just fix that quickly. I think that for some reason the video is not playing entirely. What happened? Okay, so something happened to the video. Okay, wait a sec. No, that's not. Okay, sorry. Wrong video. Anyway, there was going to be some really cool tracks, but I don't know what happened with the video. It's a different version of it. Anyway, but importantly, beyond bioimaging, we've seen on Twitter a lot of people use Napari for other purposes, and I think that's something I'm very excited about, because it shows that what we designed with Napari is not limited to the bioimage field, but potentially is something Napari can actually impact disciplines beyond biology and into physics, into remote sensing, astronomy, and many other disciplines that we are seeing on Twitter, like people using Napari for things that are very different from what we originally intended Napari for. But we always had in mind the fact that Python is not used used by scientists in many disciplines, right? And of course not just by biologists. And so when we designed Napari, we always had in mind maybe other disciplines will also find it interesting and use it. And this has been going on, that's kind of worked. So we see people using it, of course, for bioimaging, different kinds of applications, but we also seeing it used, for example, in astronomy. Here is people from YT Astro are actually thinking of developing some plugins for Napari and looking at their kind of astronomy data. We also have people like Martin, playing with nice, data sets from astronomy from the Gaia Sky project, and doing some very nice animations. We're also thinking of using that for the purpose of actually a single super resolution data and based on single molecule detection and so on. Material sciences here, for example, people using that, also starting to play with Napari for in other fields, and using kind of all the features that we developed for bioimaging, but that are to a large extent universal to images and overlaying data and processing the results of analysis on top of this data and so on and so forth. As I said, so like other disciplines, more neuroscience oriented. And even applications that have nothing to do with science, but more perhaps with art and conservation. Here, for example, the ability to open very large 2D images and browse through them using a very natural pan and zoom interface that Napari is well known for. So yeah, that's it. Thanks for your attention, and thanks for the community, for the Napari community, for this amazing work. I think most of what I've showed is not my work; is really work of the community. It's one and Nick and others at the beginning. We just had a dream, and the community made it reality. And I really thank you all for that, and thanks again for your attention.
Thanks so much, Loic. I think that we have a few questions, and so we should just probably go to that. David, you had posted a question on chat. You want to say it?
Sure. I just had I was curious about all the annotations that you're generating in Napari, how you persisted in between sessions. So what the mechanism is.
It's all NumPy, right? So in the end, when you create a layer for a given type, you basically have implicitly created an array, and that array is accessible through the scripting window, but also from the JPrimitive notebook, from where you started Napari, or from your program or whatever script that you use to start Napari. And you can always access that array like any other NumPy array, and save it yourself. But if you actually so I'm not sure if Napari is actually able to fully. I don't think so with images, with large images, we can do lazy loading, right? So we can access these very large images and just display a part of it. I'm not entirely sure we can right now. I think there are already some solutions, but I think this is an area which is, I think, evolving right now, whether you can actually, for example, make label annotations on extremely large data sets where the label image itself is very, very large, and then actually have some of these tasks as ZAR-based approaches used to actually directly persist that on the disk on the fly in a seamless manner. So I think that's possible, but that there might be a few caveats there. But in many cases, the annotations are not necessarily so large that they require to use the big guns of ZAR and desk for that. Well, depends on the application.
Loic, can we get out of the screen sharing mode so that we can see the participants? Sure. That's fine. We might have to go back into the screen sharing mode if they ask questions that. And I wasn't monitoring 100%.
I cannot for some reason access my screen. Oh, okay,
there you go.
Okay.
So I believe Davis beat Philip, but I'm not quite sure, but we'll let him go.
Okay. So do you have any idea what the roadmap is for supporting like really big chunk data, like multi-terabyte EM data? Because that currently doesn't work with Napari.
Yeah. For view, the multi-scale aspect, right?
Just the chunked aspect, because if I remember correctly, Napari is lazy with respect to Z, with respect to the plane that you're slicing, but within that plane, it tries to load everything.
Yeah,
so some of our data sets, a single plane is multiple gigs, and the data set is chunked into cubes. So in principle, if you've used Neuroglancer, that's basically like, when will Napari have the usability of Neuroglancer for these big data sets?
Yeah, I think that I think there's the working group for that, and it's definitely something that's high priority. But I can't give you any kind of concrete timeline now. It's not a trivial problem, and so yeah, but the motivation is there. I mean, the wheel is there, but we're not there yet.
Okay, Philip.
Can you hear me?
Yes.
Hi, sorry. It looks like an exciting system. You mentioned it was built on top of VisPy, and I believe VisPy supports GLSL shaders directly. How would that work within Napari?
Well, you can hack things together. Actually, this kind of galaxy visualization that Martin Wenger did was actually he played a little bit with the shaders there. And it's Python. You can basically access some of the internals of the layers. We don't recommend it, but right now, but you can, if you hack a little bit, if you can find a way, you can actually, change some of the shaders and basically, make some interesting changes there. We've been discussing whether we want to expose some sort of shader interface to let people play with shaders for rendering the different layers, but we think that the audience for that are people that would find their way in the code and be able to do just navigate through the hierarchy there and just make the changes directly. You don't have to necessarily change the code of Napari, you just have to access the right places and do the right changes. So that's possible. Yeah.
Great. Mark?
Thanks again, Loic, for this talk, and thanks for putting me on that slide, or maybe Callie is the one to thank for that. Although I don't think I've barely done anything, I feel. I think with your experience on using this kind of add or near acquisition, I was just curious on what your workflow looks like in terms of getting it from the camera into a Dask array that you could use with Napari.
Yeah, so we're actually working right now on kind of re-engineering our whole acquisition systems from the ground up, everything in Python. And I think there's quite a few efforts like that. I think Tali has been interested in working with linking Napari with Micromanager and basically eliminating any Java code on the way. Directly interfacing with the kind of Micromanager drivers that are in C++. And so I think our goal is for the first we're conducting here, my team is really to make sure we can get the data from the camera straight to Zara as fast as possible, right? And we're really working on that right now. And it's not very complicated. There's some things to pay attention to, but overall it's I think it's pretty straightforward. Yeah, there's an issue with Python Python is fundamentally not very multi-threading friendly for a number of kind of original scenes that were committed by the designers of Python early on. So that's a little unfortunate, but I think yeah, we're trying to find a way to do that. We haven't really solved all the issues, right? But yeah, it's a bit of a challenge, but we think we can get to the performance we want. I mean, basically, I don't have the answer yet, but I hope to be able to have it soon. Yeah. Do that.
Mm-hmm. Yeah, so how does it currently work? I think you said the Zara thing was kind of in the future.
Oh, so I mean, right now we have two microscopes. One microscope is still running a Java-based software I've wrote when I was postdoc, and that I have my own kind of raw format that's compressed, and it's not Zara. And then the new microscope is still using Micromanager and has basically it saves the data in TIFF, and then we have in our tool decks we have ways to convert from TIFF to Zara. So we are in the process of trying to get rid of that and do everything in Python and save straight to Zara. But we haven't really finished that project. So that's just, yeah, unfortunately, I don't have I can't tell you like what's the best way of doing it right now. So my team is working on it. So
I think something I've encountered with trying to save either Generation 5 or Zara, especially in like Windows acquisition systems, is there's an issue with creating a lot of files, and especially on Windows, the file system API is about 10 times slower than it is on Linux. It's microsecond range, but on the scale of Zara, that's a problem.
Yes. If you have millions of files and if you have millions of files and then your cost per file is non-zero, then automatically you have a problem. That's why it might be necessary to save unchunked ZARs or poorly chunks, or like ZAR files that are not, ZAR storages that are not fully chunked as much as we would like to have them. But I think the ZAR team is working on sharded star files that essentially combine different chunks into multiple chunks into a file to kind of reduce the pressure on the file system with the number of files. So yeah, I mean, as I said, we have not fully solved that problem yet.
Right. Yeah, that's the sharding discussion is something I've looked into. I think participated in with Davis's help a bit. But yeah, I think that's an issue. I think there's possible solutions there. One thing I've looked at is kind of backfilling the OME and GFF specification into H5, and then using H5 as a shard, since there's potential for chunks in there. And then basically your H5 chunks can become your star chunks, if you use the correct compression method. And there's a few in there that are compatible between the two. So, but yeah, this is something I'm thinking about too with the Color Lab. And so it would be interesting to talk about that. And I know others are. In the
end, I think you have to be pragmatic, and have the simplest possible solution that has the best possible performance. Because if you build something for a specific microscope, even if it's like 2 or 3 of them, is it worth to make something super general? Perhaps not. Perhaps what matters most is that it actually works. And maybe that might mean not saving to ZAR directly if it's really not possible to do it in a way that is simple and convenient and effective at getting the project done, right? So that's probably the only thing. There's like that. In the end, if I can use ZAR to save directly our data from camera to ZAR, then that's great. But if I can't, if I have to pay some price that I'm not willing to pay performance-wise, I'm not going to do it. It's going to be sad, but then there are some priorities there. So that's maybe the only thing that I can say is that
What's the performance issue with saving to ZAR? Because if you can save tips or you can save your own binary format,
the number of files.
But that's a degree of freedom. You get to decide the chunking. It can still be ZAR with one chunk per.
But then, yeah, you could do that. That's one way to do it. But then you actually make it very difficult to browse your data interactively with Napari, for example, because your chunks are huge. So you can't. Right now, we can really, once we convert to ZAR, we can literally browse through our data set super nicely, and that's cool. But if you have, we typically chunk more in Z than in XY. We chunk more in, of course, we chunk everything in time, so we can move in time very quickly. When we land on a specific Z slice, we only have to load a few planes around that, so it's very quick. If we had to load the whole data set, the whole stack for the whole time point, every time we hit a slice, then it would be very slow. And then so those are the trade-offs. So I think you could do, as I actually mentioned it, you could sign with ZAR with poor chunking or with no chunking, but you're gonna pay something else somewhere. You're gonna lose some of the features of ZAR essentially. Yeah. And that might be acceptable if that's just, if the raw data needs to be accessed once to process it the first time into something else that then can be, chunked appropriately for processing and visualization.
Thanks. Okay, we got a little detour into data engineering there, but Adam, you had a question?
Yeah. So maybe this is a trivial issue, but I guess I had a question about naively I thought that that basically Napari was a tool to be used within Python, and it looked like there's some instructions on the web for installing it with pip, but then you showed that sort of built-in Ipython console, and so I sort of wondered and also sort of a standalone downloadable installer for Napari. So I guess I wanted to ask sort of, do you think of Napari as mostly being sort of something that embeds Python, or do you think of it as being something to be used within a Python install, or is it usable either way, or does one of
I
those
think
have?
it's usable in many ways, and I think that's a good feature. You want something that is used that is not usable in. I mean, using it from a Jupyter notebook, it's fantastic, right? I mean, if at least if you can if you can actually run it in the machine that you're actually sitting in front, and running it standalone could be fantastic too. Of course, the problem there, you need to embed Python and you need to build to potentially update packages. There's lots of technical issues there that we need still to completely resolve, and we're discussing that actively. Actually, I was just seeing, putting some messages on GitHub, and yeah, I think I don't envision a single use of Napari through a single kind of interface. Like it can be, people might use it, within their scripts. They could be using it from within their tools. They could be using from a Jupyter notebook. They can use it from a standalone app. Yeah. So lots of, and I think it's great.
So if you launch it within like a Jupyter notebook, and then you open the built-in interactive Python console, does that open up
just opens another window? So if you're sitting on the machine that actually runs the server, that's important.
Yeah.
Then from within the Jupyter Notebook web interface, you can open a Napari window that has nothing to do with the browser that you're interacting with. It's just separate, and so you can just keep doing your work with Jupyter Notebook, and on the other side, you actually have the Napari window that you can then essentially remote control and do things with it, both from the Jupyter Notebook, but also directly on the interface. And you also can open the Ipython terminal and interact with any object that is already loaded there.
Loaded in
that's all
Napari.
part of the same Python process, so it's all.
Oh, I see. Okay.
So all the same part.
Okay.
It's the data is there. You can do everything you want to do.
Okay.
That's a popular way of interacting with Napari. But the caveat is you need to be on the machine. The server running Napari must be in the machine that you're sitting in front, right? Or maybe you can do a remote interface, but you cannot open a Napari window on a remote Jupyter server, right? Because that's going to open a Napari window on the machine you're not in front of.
Yeah.
But otherwise, it's a great.
Do you guys have any plans to like use WebAssembly or something to support Napari within?
There've been discussions about it, and maybe I think why not? I mean, it's all about like who in the community is going to be motivated to work on that. Whether some of supporters of Napari such as CZI are interested in developing that, or others. So this is really something that
So you're not that interested in developing that?
Personally, myself, no.
Okay.
But the community is the boss here. Like this is why this project is so exciting, is because it's community driven, and there's lots of things happening there that we don't necessarily have thought about.
Thanks a lot. Yeah, great talk.
Oh, you're welcome. Thanks.
So any other questions? I guess, Loic, one question I had was, when you're using Napari, are you primarily using it on like your MacBook Pro, or do you have everywhere? So like you have
I use it on
big
my
old
desktop machine, on my laptop. I've seen people use it on the Raspberry Pi. Apparently it's possible. We use it on Windows, on the scope machines that acquire the images from that control our alleged microscopes. I think I see a Napari window almost open on every of my team members' screens. So almost always. So it's really, we use it all the time on all sorts of different machines. And it also works on the M1, which is the machine I have here.
But if you're working on really large scale image volumes, is there kind of like a minimum platform that you wind up using?
So we try to have 10. Yeah, so we try. So we have most of our data is in the server. Like a big server with like petabytes and petabytes of data there. And we have 10G to 100G connections to our desktop machines. that let us browse the data remotely with Napari.
I see.
And that works. There are, of course, performance-wise, like it's kind. I mean, our data is huge, so it just gets bigger and bigger, yeah. So it's a bit challenging, but it does work. And the more you process your data, the smaller it gets, and sometimes the compression can get more effective the more you process your data. And so sometimes, the final product can be a 100 gigabyte file that came from 20 terabytes of data, with 100-fold compression sometimes, and then you basically then it's very, very easy to go through the data even on a laptop.
When was the last time you used Fiji?
I do use it, maybe once every 2 months. Fiji is the way I understand. Of course, increasingly less so. I'm using, of course, Napari is fitting a lot of our use cases, but there are situations where, you need to open the data set that's in the Word format, or sometimes I really need to go back to the pixel in its primordial form. And my first exposure to voxels was in Fiji. So of course, I have to go back to the basics sometimes, like, okay, what is this image really looking like, independently of that fancy tool that we've developed, right? And so sometimes I have to go back to, yeah. But yeah, I think yeah, it's Fiji is a great tool and has a lot of users, and we see a lot of very nice efforts to bridge the gap between the two communities. Robert has done a lot of work there, and others too. So it's very exciting.
Okay, great. Thank you very much, Loic. And thanks for being generous with your time.